Dear audience, thank you for being here. I would like to show some aspects about our wastewater-based techniques and results. First of all, the municipal wastewater is a gold mine because it contains all substances that are excreted from the body with feces, urine, or washed with household water into the sewer system. Fortunately, in Hungary, the majority of the population is connected to the centralized sewer system, so we can obtain a comprehensive overview from the health status of the supplying population with a representative sample from the wastewater treatment plant, of course the raw wastewater. This technique started in the 1940s when wastewater-based epidemiology was applied for the detection and distribution of poliovirus in the sewage and was used for some chemical investigation in the early 2000s. Nowadays, we can examine a lot of different targets from infections and non-infectious diseases to different metabolites and biomarkers, as you see in the bubbles. Wastewater-based epidemiology gained wider interest during the COVID pandemic as a non-invasive, non-contactive, population-based way which supplement the traditional methods. And in the case of SARS-CoV-2, it can also be used for forecasting. In Hungary, we start the work with wastewater in the spring of 2020, but the weekly sampling frequency start from July. We have some regular sampling points, like the 3 wastewater treatment plants in Budapest and other treatment plants in country seats. We have one composite sample from Budapest agglomeration and one sample point in Budapest airport. It means our samples cover approximately 40% of the national population. The sampling is the task of the operators of treatment plants, and government offices are liable for the delivery of the samples, which processed within a day in our laboratory. We focused on molecular nucleic acid-based methods. The processing contains 3 easy steps, like concentration, nucleic acid isolation, and detection and evaluation, but in fact the reality is not that simple. So this flow chart shows our standard design pipeline. The upper side contains the chemical examination, and the right side shows the poliovirus pathway, which was presented earlier by my colleagues. So I want to focus in the middle part of this picture. So after the centrifugation, bacterial and unicellular parameters are examined from the pallet, and we investigate enteric pathogens like Enterococcus, Campylobacter, Salmonella, Clostridia, and Cryptosporidia, and some antibiotic resistance genes from wastewater with qPCR. In parallel, after the centrifugation, viral parameters are analyzed from supernatant after an specific ultrafiltration, and SARS-CoV-2 and influenza A analyzed with digital PCR and sequencing methods, and other respiratory and enteric pathogens analyzed with qPCR. And if the epidemiological circumstances needed, we can examine other targets like Mpox or dengue virus or Bordetella or other pathogens. In the first time, during the first waves of SARS-CoV-2, qPCR was enough for detection and forecast. As you see, the trends of wastewater forecasted the trends, the number of new cases 1 week earlier, and 2 weeks earlier the patients recurring in hospital treatments. Later, the alpha variant was appeared, and we could follow the spreading of this variant with different melting point PCRs and digital Doppler PCR also. Nowadays, we quantify SARS-CoV-2 with digital PCR and normalize with peppermint model virus. We do a lot of statistical analysis, and we quantified Omicron variants with next generation sequencing. Over the standard methods, we can adapt our methods quite quickly, so we were able to measure Mpox, Bordetella pertussis, and dengue virus when needed. And at the end of 2024, human influenza data started to rise, but the measured influenza A concentration from the wastewater did not show this tendency, so we wanted to know what is in the background. And yes, human cases occurred by influenza B, and now we examine weekly influenza B also. So our investigations are still ongoing. We have respiratory panels which reported every week, but not only the results from the wastewater, because we always comprise the human and wastewater data in an integrated respiratory surveillance report. We joined some international project and proficiency test for SARS-CoV-2 in last November. Or other targets like enteric pathogens or antibiotic resistance are not published yet, but there are some current results. And to work with chemicals is a future aims, but we join an international drug survey last May. The sampling point was the central wastewater treatment plant in Budapest, and as you see our results, the cocaine and ecstasy use was higher in weekends, and as you see, the marijuana use was quite similar during the week. We know this method has a lot of limitation. We have little information sometimes on the extraction of the targets or shedding or decay kinetics of that. We show the detection threshold rates are very variable, and the environmental factors has very strong effect, and the timing is also essentials, and we also fight with the inhibitors in the wastewater. But we know that the wastewater is a very good baseline. The material, the genetic material is still present in the wastewater always, and for several pathogens, this method is very good for the production. In Hungary, the network and logistics system is built up, and we have opportunities to measure smaller locations, also like airport, quarantine hospital, refugee camps, or, for example, in the BSL laboratory wastewater. And the main point that this method can fast adapt for a wide range of targets from community nucleic acid. Me and my colleagues would like to thank our colleagues from National Center for Public Health and Pharmacy, and partners from wastewater treatment plants and from government offices. And thank you for your attention.